The fibroblasts are early responders to injury and they are the key player in the wound healing. These are collagen secreting cells that build and maintain the extracellular matrix, and they are actually activated by a biochemical environment that is rich in cytokines and growth factors such as platelet derived growth factor, fibroblast growth factor, and transforming growth factor beta. They are involved in all stages of repair. In the inflammatory phase of the repair, they are recruited to the injury site in response to the chemotactic signal, and they are activated to begin extracellular matrix. Then in the proliferative phase, they proliferate and migrate into the wound and synthesize collagen, fibronectin, and other extracellular matrix protein. In remodeling phase, they participate in extracellular matrix remodeling and scar formulation, and they regulate balance between extracellular matrix deposition and degradation. They are also capable to differentiate into myofibroblast under the influence of cytokines such as transforming growth factor beta. These fibroblasts differentiate into myofibroblast. The myofibroblast have got actin stress fiber that contain smooth muscle actin alpha, and they have got contractile ability. They pull the wound edges together and aid in wound contraction. Bone marrow also contributes into the wound healing. Some of the bone marrow derived mononuclear cell can adopt a fibroblast-like phenotype. These mesenchymal stem cells and fibrocytes are examples. They migrate to the wound and transiently contribute to the repair. The fibrocytes are thought to have a role in fibrosis and scar formation, and despite their recruitment, these marrow-derived cells do not become permanent resident of the local connective tissue. The fibroblast and myofibroblast are central to healing, but also to they lead to pathological fibrosis when the extracellular production, matrix production, and remodeling go unchecked. For example, in liver, lung, and kidney, it induces fibrosis. Then these cells are also therapeutic target, because if you understand the processes that has driven the development of and differentiation of these cells, you can go towards the antifibrotic therapies in chronic inflammatory diseases. So this image is providing an electron microscopic view of chick embryo fibroblast, and this fibroblast F is embedded in a collagen-rich extracellular matrix environment. So these fibroblasts are elongated, spindle-shaped cells with, this is the elongated spindle-shaped cell with a dark irregular nucleus. The cytoplasm has got these blackish dots. These are actually scattered ribosomes, and it is showing a rich granular endoplasmic reticulum. There is presence of these fibroblast extensions that are reaching out towards the surrounding matrix. The collagen fibers appear as cross-striated structure. In some areas you can see these are cut transversely, and they are depicted as these black dots, and longitudinally as wavy fibers, these wavy fibers that are lying parallelly. The electron dense matrix here, these are the collagen fiber, dense organized matrix formed around the fibroblast. It is not uniformly dense that reflect the dynamic extracellular matrix remodeling by the fibroblast. So this is clearly showing the interaction of a fibroblast with surrounding collagen matrix, highlighting the active role of fibroblast in collagen production and organization of extracellular matrix, and the close packing and orientation of the collagen fibers typical of developing or repairing tissue. The cell's secretory and synthetic profile is reflected by the presence of organelles like rough endoplasmic reticulum. This picture is also showing a fibroblast that has got a nucleus, and over here this prominent central nucleus has got a well-defined nucleolus that is typical of active or metabolically engaging cells, and there is active cytoplasmic reticulum also, and abundance of this rough endoplasmic reticulum that surround the nucleus, that is surrounding the nucleus, and is responsible for formation of intense protein synthesis, that is production of collagen and extracellular matrix by these fibroblasts. Okay, then there is presence of these bundles of the collagen that are depicted by B, and they are reflected by these black dots in cross section, and some are wavy fibers. These are blackish dots of cross sections of the fibrin of collagen, and some wavy fibers of the collagen that are longitudinally cut. So these are the newly synthesized and secreted collagen. Then there are fibropositors that are highlighted by these arrows, the fibropositors. These fibropositors are actually the extensions of the fibroblast cytoplasm that surround and guide the fibrils of the collagen. These are the specialized structures through which the fibroblast orchestrate the assembly and extrusion of the collagen fiber directly into the extracellular matrix. Some of these newly formed fibrils are seen enveloped by the fibroblast membrane and cytoplasm, indicating the extracellular assembly and export of the collagen. The biological implication of these are there is active extracellular matrix synthesis. There is central role of fibroblasts in the production of extracellular matrix, collagen, and other structural protein. They have got dynamic interaction with extracellular matrix, and they do not simply secrete the collagen. They actively organize the collagen through fibropositor, contributing to the precise extracellular matrix architecture that is critical for the tissue repair and integrity. Also, they are responsible for healing and tissue strength because this process is fundamental for wound healing, fibrosis, and remodeling of the tissue. If the fibroblast properly function, that will work as tissue regeneration, or production of the collagen after the injury. If there is dysregulation in this process, it will lead to fibrosis or pathological effect of fibrosis in the organs such as lung, liver, or heart. This set of images display a series of histologic and electron microscopy images of fibroblast and myofibroblast within a tissue sample, and these are stained. The electron immunohistochemistry stain that is used in this is smooth muscle actin or SMA, which highlight myofibroblast. All three magnification 400, 40 and 1000x are used to highlight the architecture as well as the individual cell. The fibroblasts are spindle-shaped cells that are lightly stained uh, cytoplasm, while the myofibroblasts are spindle-shaped also, but with a darker, more intense brownish cytoplasmic hue or color that is due to smooth muscle actin positivity. So they clearly differentiate the fibroblast and myofibroblast, the fibroblast and myofibroblast on the basis of immunohistochemistry. The electron microscopic picture, this picture is showing the fine cellular detail. There is dense bundles of actin filament that are highlighted by these arrows, and these are the hallmark for their contractile phenotype. The surrounding extracellular matrix and these black dots or collagen fibrils are also visible. These images are from the site of fibrosis or wound healing that show fibroblasts transform into myofibroblast to produce and remodel the extracellular matrix. The myofibroblasts contribute to wound contraction and scar formation. In chronic injury, their persistent activation can lead to fibrosis, pathologic fibrosis and liver cirrhosis, uh, lung fibrosis, etc. So this composite of light microscopy, electron microscopy illustrates the abundance of myofibroblast in that area, the ultra-structural feature that is actin bundle for contractility, and their role in the tissue remodeling and repair. In this image, the differentiation of fibroblast into myofibroblast and the role of transforming growth factor beta 1 and the mechanical tension in this process is illustrated. So here you can see that there is a fibroblast, and this fibroblast is an elongated cell with a central nucleus that is surrounded by a network of fibronectin. These green threads, these are fibronectin, these green threads, these are fibronectin, and there is presence of cortical cytoplasmic actin, that is this orange area. So no prominent alpha smooth muscle actin is seen at this stage. Then, under the influence of mechanical tension, the proto-myofibroblasts are formed that is an intermediate stage between fibroblast and myofibroblast. So the mechanical tension and transforming growth factor beta 1 signaling stimulate the development of alpha smooth muscle actin containing stress fibers that are these pink lines. These stress fibers are formed under the influence of mechanical tension and transforming growth factor beta 1. So these pink lines are developed, and these are still connected to extracellular matrix by focal adhesion. These pink ovals, these are focal adhesions. These focal adhesions. Through this, they are connected to the extracellular matrix. Then there is formation of differentiated myofibroblast. This differentiated myofibroblast is formed under the same stimulus, and it is a fully developed myofibroblast with prominent alpha smooth muscle fiber across the cytoplasm. The focal adhesions over here, they anchor the stress fibers, actin stress fibers, to the extracellular matrix. These are the EDA fibronectin or the fibronectin that are present in the extracellular area, and they provide contractility. EDA fibronectin, that is orange one, and the fibronectin, that is green, highlighting the extracellular matrix component that is involved. Over here, this column is showing the transforming growth factor beta activation pathway. Here, the latent transforming growth factor beta 1, it is produced by a cell and secreted and bound to latent transforming growth factor beta binding protein. And then the latent transforming growth factor binding protein with transforming beta 1 complex is formed, and this complex binds to extracellular matrix protein such as collagen, fibronectin, and fibrillin. Integrin receptors on the cell surface bind to this complex. The mechanical stress or tension that is exerted by the cell via actin stress fibers causes a conformational changes in the lab or latent actin latency associated protein, and this liberates active transforming growth factor beta 1, which can then promote differentiation of fibroblast and remodeling of extracellular matrix. So mechanical tension provides the stress fiber in the cytoskeleton of the cell, and it is essential for transition from fibroblast to proto-myofibroblast and then differentiated myofibroblast. The alpha smooth muscle actin and focal adhesions are critical for contractility and stabilization of the myofibroblast, and integrin in the focal adhesion link the actin cytoskeleton to the extracellular matrix proteins such as collagen and fibronectin. The fibronectin and EDA fibronectin provides a provisional matrix for fibroblast attachment and differentiation. So in this whole process, the transforming growth factor beta 1 is the central regulator of myofibroblast differentiation, and it promotes synthesis of smooth muscle actin, extracellular matrix protein like collagen, and fibronectin. So these myofibroblasts are essential for the contraction of the wound and scar formation. However, if they are persistent or pathologically active, they may lead to fibrosis in the vital organs. Thank you for watching this video. If you have any query, please write in the comment box.